This video explores the genetic landscape of North India and Uttarakhand, focusing on historical migration, sex-biased admixture, and the genetic patterns of scheduled caste and scheduled tribe populations in the Himalayas. A notable phenomenon is the elevated presence of R1a lineages in SCST populations, paired with the dilution of the deeply indigenous haplogroup H, R1a-L657 and Z2124 are linked to Bronze Age migrations, while Haplogroup H is associated with ancient hunter-gatherer roots. These markers represent distinct histories. Haplogroup H is deeply rooted in the subcontinent. In contrast, R1a subclades trace back to steppe pastoralists. In Uttarakhand, however, we see an unusual elevation of these R1a markers within SCST communities. 4 processes drive this: sex-biased admixture, the integration of the Khasas, specific founder effects in isolated mountain terrain, and the fluid nature of tribal labels which encompass diverse ancestral groups. Sex-biased admixture played a key role. Through anuloma marriage patterns before rigid endogamy, R1a was introduced into indigenous lineages. Because Y-DNA is paternally inherited, these markers persisted even as subsequent generations identified with the mother's community. The Khasas were central to this history. Unlike the rigid hierarchies of the plains, hill society remained fluid. This allowed for symbiotic coexistence and extensive mixing, leaving a legacy of high steppe-related ancestry in modern mountain populations. Geography also acted as a funnel. Rugged Himalayan terrain isolated small groups, allowing genetic drift to amplify the R1a marker. Over generations, this significantly reduced or replaced older indigenous lineages in many communities. Finally, the label Scheduled Tribe covers diverse origins in this region, from Tibeto-Burman groups and Terai populations to stratified Indo-Aryan groups like the Jaunsaris, who carry higher frequencies of R1a. The takeaway is that modern social labels do not equate to isolated biological groups. The patterns we see today are the result of a deep, shared history of migration, interaction, and gene flow across the Himalayan region.